Today, our discussion will be about renal physiology. First of all, we will talk about the distribution of fluid inside our body. If a person has a body mass of 70 kg, so that person will have the 60% of water. The total body water will be 60% of the body mass, like it will be 42 liters or 42 kg. All right, the non-water mass will be 40% of the body mass, that will be 28 kg. So this 60% of body mass, that is 42 liters of water, will further divided into two portions of our body. The 40% of this fluid is present in intracellular spaces, or this fluid will known as intracellular fluid. The other 20% of this will present in extracellular spaces, that can be interstitial fluid or plasma. So high potassium intracellularly, the potassium will present in high amount inside the cells. There is a rule 60-40-20. So the percentage of body weight for average person, the 60% of total body water in every person, the 40% of which is intracellular and the 20% of which is extracellular. The plasma volume can be measured by labeling albumin. As we know, the plasma inside the blood vessels is carried out through albumin. Albumin is a molecule that is used for the transport of plasma. There is an oncotic and hydrostatic pressure present inside our blood vessels that is handled by albumin. The extracellular volume can be measured by two substances. The one is inulin and the other is mannitol. The osmolarity is 285 to 295 milliosmol per kg of water. There is a mnemonic used is MIS. So there are four substances: mannitol, inulin, sodium, and sucrose. These substances are distributed only in extracellular spaces like intravascular and interstitial spaces, not within the cell. Okay, let's a few words about glomerular filtration rate. So the inulin clearance can be used to calculate GFR because it is freely filtered and is neither reabsorbed nor secreted. The GFR is equal to urine concentration of inulin cross urine flow rate over plasma concentration of inulin is equal to clearance of inulin from the urine. Yes. So the normal GFR is 100 ml per minute. So the creatinine clearance is an appropriate measure of GFR. Slightly overestimates GFR because creatinine is moderately secreted by renal tubules. So the incremental reductions in GFR define the stages of chronic kidney disease. The graph between plasma creatinine and glomerular filtration is not linear. So here is a graph that is non-linear. GFR is essentially normal in a patient or when the 50% of the kidneys are working. So when there is a decrease in the GFR of below 60, so when the GFR is below 60 instead of 100, so that at that point the creatinine is a best source to measure the GFR of a patient. So when there is a decrease in GFR below 60, then the creatinine will raise, and that creatinine can tell us the actual reduction in GFR. So if the GFR halves, the creatinine will double after 60 ml per minute. So effective renal plasma flow. Effective renal plasma flow can be estimated using para-aminohypuric acid clearance. Between filtration and secretion, there is nearly 100% excretion of all para-aminohypuric acid that enters the kidney. So here is the same formula. Effective renal plasma flow can be measured by using this formula. Renal blood flow is equal to renal plasma flow over 1 minus HCT. HCT is hematocrit. So plasma is equal to 1 minus hematocrit. Effective renal plasma flow underestimates true renal plasma flow slightly. So there is a slight underestimation of effective renal plasma flow. Let's talk about the filtration of kidneys, or filtration fraction is equal to GFR over renal plasma flow. So normal filtration fraction is 20%. Filtered load is equal to GFR cross plasma concentration. GFR can be estimated with creatinine clearance, as we had discussed it already. So renal plasma flow is best estimated with para-aminohypuric acid, as we discussed already. So prostaglandins dilate efferent arteriole PDA, because in patent ductus arteriosus there is a function of prostaglandin, as you already studied in the CVS or cardiovascular system, or we will study this topic there. And ACE inhibitors constrict efferent arteriole. ACE, they constrict efferent arteriole by the angiotensin 2. So we will discuss about that mechanism later, how the angiotensin 2 constrict efferent arterioles. So as you can see in this picture, the blood enters from the efferent arterioles and the filtration process occurs in this space in the glomerulus. Here are three components of glomerulus. The first is fenestrated capillary endothelium. The second is glomerular basement membrane with type 4 collagen, and the third is podocytes. So all the filtration occurs here, and the remaining blood will pass out from the efferent arteriole through this way. So when this reaches to the proximal tubule, so if there is any reabsorption or secretion, and if there is any substance that need to be secreted, so that will secrete back to the proximal tubule, or if there is any reabsorption, so that will happen through this pathway. So here is a very short discussion about filtration. So as you can see, the efferent arteriole angiotensin 2 constrict these efferent arteriole, that will help in reduction in renal plasma flow and increase in glomerular filtration rate. So there will be increased filtration fraction. So while on prostaglandin dilate efferent arteriole, that will increase renal plasma flow, increase GFR, so no change in filtration fraction. So changes in glomerular dynamics. When efferent arteriole is constricted, that will lead to decreased GFR and decreased renal plasma flow. So as you can see here, we have efferent arteriole. When this is constricted, so what does that mean? There is decreased amount of blood flow to the glomerulus. So when the decreased amount of blood flow, what will happen? There will decreased glomerular filtration rate. So if the efferent arteriole is constricted, if the efferent, here we have efferent arteriole, if this is constricted, what will happen? So it's like a pipe. When this is constricted, there will be increased amount of blood before that constriction. So what is that before? Here we have a glomerulus. When there is increased amount of blood, that means the glomerulus can filter more blood. So there is increased GFR in constricted efferent arteriole, and there will decrease renal plasma flow. So that will lead to increased filtration fraction. When there is increased amount of plasma protein concentration, that will lead to decreased GFR. When there is increased amount of plasma protein concentration, so as we know, the proteins cannot easily filtered out through glomerulus, so that will lead to decreased GFR, and this will lead to decreased filtration fraction. If there is decreased plasma protein concentration, that will lead to increased GFR, and that will lead to increased filtration fraction. So when there is decreased amount of plasma proteins present in the fluid or blood, so that means proteins, the glomerulus cannot filter the proteins, so the glomerulus will get enough time to filter the other products, so there will increase GFR. When there is constriction of ureter, so if the ureter is constricted, it means the outflow pipe is constricted. That means the urine cannot pass through that much, so that will put the backup pressure on the glomerulus, and glomerulus cannot filter all the plasma because the outgoing pressure or outgoing space is not enough to handle all the upcoming filtrations. So that will lead to decreased GFR, and that will lead to decreased filtration fraction. So when there is dehydration, that means the blood flow to the kidney is very less, as in dehydration it usually happens. So that will also lead to decreased GFR, and there will be very decrease in renal plasma flow, and there will increase in filtration fraction. So with acute renal obstruction, first 12 hours, the renal plasma flow may transiently increase. However, with time, the efferent arteriolar constriction will lead to decreased renal plasma flow in response to reduced GFR. Even at later stages, though the GFR remains depressed to a greater extent than the renal plasma flow, resulting in reduced filtration fraction. So the very next section is about glucose clearance. So glucose at a normal plasma level range 60 to 120 milligram per deciliter is completely reabsorbed in proximal convoluted tubule by sodium glucose co-transporter. So this is the transporter that reabsorb all the glucose from the proximal convoluted tubule. So if in adults at plasma glucose of ranging in between 200 milligram per deciliter, so that will lead to glucoseuria, and at rate of 375 mg per minute, all transporters are fully saturated at this concentration when there is a 200 milligram per deciliter of glucose is present in the plasma. All right. So during normal pregnancy may also decrease ability of proximal convoluted tubules to reabsorb glucose and amino acids. So that condition will also lead to glucose urea and amino acid urea. So sodium glucose co-transporter to inhibitors like flourishing drugs permit glucose urea at plasma level concentration less than 200. So the threshold of glucose urea will decrease to less than 200 in some drugs. The glucose urea is an important clinical clue to diabetes mellitus. So SPLA is a region of substance clearance between threshold and TM due to heterogeneity of nephrons. So as you can see, here is a normal diagram about the plasma glucose and glucose transport in this condition. Okay, so the threshold is 200, renal threshold, that will lead to glucose urea. So when it is in normal condition like 160 to 120, so there is a filtration is coming that way and there is no problem. When this threshold is passed, so that will lead to a SPLA and there will be increased amount like 375 mg per minute, because this line is crossed, so that is the maximum. When there is increased amount, so all transporters are fully saturated at this condition, so the other amount of glucose will not be reabsorbed, so that will be excreted in the urine.